Thank you for opportunity to talk to all of you. First, I will just say a few words about who we are. Genus is animal genetic company. All what we do, we breed in animals and we distribute genetics to many customers, farmers around the world. Our pork division is called PIC, Pork Improvement Company. It's our business unit. And our dairy and beef is ABS division. And I'm pretty sure that there are quite a few people who are aware of PIC or ABS. And in our company, we also have R&D organization, which is really shared platform, technology platforms, and R&D works on things like gene editing, genomic science, reproductive biology, data, etc., and sorting semen. And we work across all the species. I think with that, I just want to jump right to what I want to talk to you today. As I was thinking about questions that were asked about genomic selection and gene editing, and also taking in consideration that we are working on commercialization of the PRRS trait. And Christine was talking about how important PRRS and why we're working on it. We do know our customers cannot wait for that trait to be delivered to them. And also Clint Nesbitt, sitting right there, would be talking more about what we do to commercialize PRRS. So I'm not talking about that. Well, instead, what I want to talk about a question that's being asked by a lot of regulators around the globe as we going into all the conversations with different countries. And the one question that they often ask us, could genetic changes that you introduce by gene editing have arisen naturally? And it's a very important question, because if they are, we will have very easy way to get through regulatory process. If they're not, the regulatory process becomes very long, expensive, and difficult. So therefore, it's really important for us to understand what changes could have arisen through naturally, right? And I firmly believe that the only way we can understand it through understanding more about genomic variation that present in our population. And the question is, how much do we know? We also heard today that Anna was saying that we need more research. Well, the question is what kind of research, right? And how much already available? And I'm coming to this animal genetics from plant genetics. So I had a really interesting time because we did with plants, because we actually did focus on that question, how much genetic variation is present across species. And a lot of was published on that topic. When I started looking and tried to answer that question for myself, I realized there's not much information out there. And not much research is being done. And then the question, maybe we already have a lot and we just don't know about it. So how we can get to genetic variation? We all know that we're using SNPs for genomic evaluations. And what SNPs are, it's actually genetic variation, right, present in our population. But it's also important that we need to understand how much we already know. So if we think about cow genome was introduced in 2007 and was individual animal genome. And once all researchers start comparing DNA sequences, they realize that there is a lot more variation exists, right, in that species. In 2009, pig genome, again, individual animal, the same story, doesn't describe all the variation, of course, that exists in the population. In around 2018, functional annotation of cattle and pig genomes came to life through FANG, and it's actually was fantastic because it started helping us to understand not only the variation, but also biological meaning of that variation. And that is critical because most of the SNPs, as we know, are not encoding regions of the genes, so it's really doesn't necessarily clear to us which SNPs lead to what biological changes, which we really need to understand. And then eventually pangenome started. And this is the era of pangenomes, which I think would be really critical for all of us, because now we're bringing together all the genomic sequence across the whole population of the species. And that's actually hopefully will help us to understand a lot more. And something else will come next. But the question is it enough, even with all these efforts? So we asked ourselves that question, and we decided to do some small experiment. What we did, we looked at public databases where all public SNPs for pigs described, right? And forgive me for a lot of numbers, but I will take you through them. For example, if you combine all the SNPs, right, and see how many unique SNPs across these two databases, well, that would be around, what's the number? I forgot, but it's over 1 million unique SNPs. So stop gain lost, which is very important, is around 8,000. Synonymous 287, and then missense, which is also important to us because it means potential impact on biological functions, is around 227. Big numbers. But then we decided to look at our own SNPs program, SNPs data. So we have our SNPs, and we also have whole genome sequences that we did around on 7,000 individual pigs. And what we found that we have around 100 million of SNPs in our own database. Well, then we compared this number to public data, and what we found that there is 37 million, well, yeah, there is a lot of SNPs present there that actually unique in our own database. So they're not present in the public database. But what most importantly, if you look at stop gain lost, it's around 14,000, and missense another 217 unique. They're not present today in a public database, right? So if we look at this number and we think about genetic variation that we use as our opportunity to say what is present in current populations, we just doubled the number of biologically important SNPs, right, between our private database and public database. So therefore, if we want to answer the question, how much variation, a lot more than we believe exists based on the public data. And that's only based on 7,000 whole genome sequences of the pigs. Well, we have millions of them, which we haven't sequenced yet. So why this number is important? Because they just showing to us how much variation actually present. But it's not enough, because then the next question is, but will any of these variations lead to any de novo variants? So what is de novo variant? De novo variant is really about genetic change that is present for the first time. So never was present before, or at least not present in the parents, right? So if you take parents, you cross them, you got your litter, how much de novo variants each piglet in that litter will have that is not present in the parents? Well, we calculated that. How we did it, we actually used our database and we looked at the crosses between male, female parents, and we looked at the FY generation, and we did whole genome sequences of only 13 liters in those trios, which is exactly important because that's how we define what de novo variants are. So it was interesting for us to see that we found 1,367 total, and it means that between 88 to 134 variants per individual, right? And out of them, 394 were indels. So it's 24 to 43 per individual. Why indels important? Because there are deletions or some insertions that actually change in the biological functioning of specific genes. So in genes itself, 740, encoding regions 37, and then missense 2. It's kind of numbers that we expected actually to see, but we never had them. So if we look at human and mouse data, where this data published, it actually fits the number. It's around 100 de novo variants per individual per generation. Now that takes me back to the question, can gene-edited mutations be arising naturally? Well, to answer that question, we actually don't have all the information, because that's what we see today. And what we see that there are hundreds of variations in coding regions. And now we need to think about, okay, what does it mean? Well, that's why functional annotation is so important, right? And the work that was done on public annotation of genomes is critical, because now we can look, what does it mean? So remember, we approximately 37 encoding regions of the gene per one individual. So if we start looking in answering simple question, well, what genes? What does it mean in this liter? What genes has changed? It's interesting. There are a lot of very useful changes. And I will give you here only one example. So in here, you see this huge deletion. Oops, sorry, I will go right back. Okay. So you see this very large deletion, for example, right? So this large deletion in coding gene, this coding gene, I call it gene 1, just for confidentiality purpose. It's actually methyltransferase. Well, this gene implicated in the replication of all enteroviruses. So we found that mouse that deficient in that gene resistant to enterovirus infection. Well, that's very interesting, because now we have naturally occurring huge deletion in the gene that can lead to disease resistance. Then you will ask, well, why wouldn't you use that and breed those animals? Well, actually, because we captured it after animals were already dead, because that was from our database. Secondly, because we actually can't do whole genome sequence on every born animal. It is expensive today, cost prohibitive. Secondly, we don't know anything about, do we need, for full resistance, do we need homozygous, heterozygous? That needs to be checked, right? And we can do it. But it was very interesting. We looked at the other mutations, that new mutations, and we found in those mutations of all sizes, in variety of functional genes that link to immune system, link to productivity traits, link to many other things. Now, can leads the question. So when question asked, can gene editing reproduce natural mutations? Yes, what we need to know, this information, right? And to produce this information, we need to do a lot of work. But I think the key question here, key conclusion here, just from very brief and short experiment, we see that nature creates millions genetic variants every reproductive cycle, right? So, and we breed animals all the time. Traditional breeding, as we heard, will, of course, make use of all this range of genetic variation, but only phenotypes observed. If phenotypes not observed, those genetic variants could be easily lost during breeding. Why? Because we don't see the phenotype. We can't test for that phenotype. We can't test easily population for disease, right? We cannot expose a lot of animals consistently to diseases to see it. Also because those mutations are present in heterozygous state. So obviously during following up crosses, most likely they will be eliminated from the population. They probably will occur again, But again, we need to capture them in time to use them. So yes, and if we found them, we can make use of them, but it will take many years. Because if we found that variant in one animal in heterozygous state, it will take us quite a lot of years to propagate that mutation through our whole population, and most likely it would need to be in a homozygous state. But, and while we're doing that, of course, right, we can lose on most important traits that we usually breeding for. And the gain on other traits could be lost during that process, which is not what our customers want from us. But what we can do, we can make that deletion in our elite germplasm today and test for the phenotype, right? But when we do that, we would be asked the question, you deleted portion of the gene, can this be, show us that it can occur naturally? Well, in this case, we can, because we have the data. But I think the problem here is that when we're thinking about that question, and forgive me for that ambitious statement, but I do believe so. I do believe that genetic changes that we can think of and introduce through gene editing most likely already occurred, or they will be in our populations where we consistently cross animals and we actually expand in the diversity of our population. So I think that's short presentation that I wanted to just bring the question for all of us, and especially considering that it is a regulatory workshop, to just challenge our thinking about questions that usually we're being asked by all regulatory agencies around the globe, and the burden of proof that we have to bring forward to prove that this gene editing change can naturally occur. And I think that's the complexity of where we are today, because some of us will have more data to show it, some of us will have less. But the reality of that there should be probably some work done across companies, across academia, to demonstrate the broad variation that exists across the species that we're interested to work with. So thank you. That's all what I wanted to share today.